All right, how's everybody doing today? Welcome to another edition of Zog Science. Today we're going to be taking a look at kind of overall systems perspective, and in particular we're going to be taking a look at developmental genetics, kind of what are some of the genetic reasons as to why we develop the way we do. So let's get started. First of all, model organisms are organisms that are easy to raise in a lab and use in experiments. And fruit flies are a great model organism. They were used in a wide variety of experiments. We've already seen them before in things like sex determination. So they were used a lot in the developmental genetic studies, which is why we're kind of talking about them. Just to prep you, we are going to be taking a look at flies today. So just a brief recap of development. Remember that fertilization occurs when a sperm meets an egg cell, and we then form a zygote or a fertilized egg cell, which is a single cell. That single cell will then divide into two cells and become an embryo. That eventually it's going to continue to divide, and it will eventually grow into an organism. You can kind of see that what happens is that as you get more and more cells, you get into kind of this ball, and then you get a nice kind of inner layer on the inside. And the inner layer on the inside actually becomes the body cavity of an organism. And we'll get more into all of this kind of developmental stuff in terms of the actual how things cellularly look when we get to the different types of animals and different species and things. But for now, we're just going to kind of talk about it from a genetics perspective. So we've mentioned the idea of differentiation before, but just as a recap, again, differentiation is when cells become specialized in structure and function. They are not randomly distributed, but rather this is a highly coordinated affair between different cells. And there are kind of two main things that are going to affect how a cell becomes differentiated. Number one are cytoplasmic determinants, and the other are inductive signals. So cytoplasmic determinants, these are molecules that are present inside the unfertilized egg cell. So that makes sense, right? They're called cytoplasmic. So they're inside the cytoplasm, but they're not uniformly distributed within the cytoplasm. There's kind of a higher concentration on one side of one type, or maybe a couple types, and then a higher concentration on the other side. So what happens is that when the cytoplasm splits during the first mitotic cycle, what happens is that your daughter cells end up having varying amounts of each of those determinants, which means that now you have different molecules in each one. Therefore, that leads to our different developmental courses. And so those two cells now are kind of going to be moving into different directions in terms of what types of cells they are going to eventually turn into. The other kinds of signals we can have are inductive signals, and this is where we have signal molecules that are released by one cell that influence the genetic expression of another cell. This is known as induction, so it's inducing a response from one cell. And this is, we've talked about this before when we did gene regulation, signal transduction pathways, where we have a signal molecule that is reduced, it binds with a receptor, that receptor then goes along and kind of sets off a cascade of pathways that eventually leads to particular gene expression. Particular genes being expressed means the cells are going to become differentiated. Now, before people kind of understood the molecular process of how differentiation works, they had this idea that, okay, we know that somehow cells become different things, but we don't really understand the mechanism how, so we're going to call this determination. So basically, before the cell is actually turned into a specific type of cell, it has already been predetermined that it will become that type of cell. And how they kind of figured this out was that they would take cells from an embryo and they would move them to a different part of an embryo. And even though they might have been surrounded by cells that were going to turn into, let's say, the head, they still turned into cells that were a part of the tail. Right, so they had already been determined. It was already predetermined that those cells would become a certain way, and so that's what determination is. You have it's irreversible, and it's due again to those molecular signals, either the induction or the cytoplasmic determinants from very early development. So this is something that gets set in place pretty early in embryonic development, probably. Before there are too many cells to really be able to see in a blastula. So this is kind of how determination works. So we've got our nucleus, right? And it's got an embryonic precursor cell, which can turn into kind of anything. all of the different genes are off. In this case, we're looking at what would become a muscle cell. So all of the muscle-specific cells and the master regulatory gene, which is called MyoD, they're all off. Eventually, it is determined through other chemicals that this cell is going to become a skeletal muscle. And so what happens is that it becomes a myoblast, meaning that is now determined. The mRNA for MyoD is being expressed, so we are forming MyoD protein, which is a transcription factor. That transcription factor will then cut on other transcription factors that will eventually cause certain proteins and other things that are specific to muscle cells to be initiated, therefore differentiating that cell into a different type of cell. So at this stage, these are still embryonic precursor cells. They haven't actually become what they will become, but however, it is already determined that they will be due to the chemical signals that are inside. So that's kind of the difference between determination and differentiation. Determination, you haven't actually become, but you know what you're going to be. Differentiation, you are now what you were set out to become in terms of what, we're talking about cells in this case. So pattern formation, okay, so that kind of gives us an idea of sort of some of the chemical things that are going on. Pattern formation is where these signals kind of contribute to a spatial organization within the embryo. So we want to know, where is the head or the anterior end, where's the tail or the posterior end, where's the ventral or front, the dorsal, back, the left, and the right. And this is very important because you want specific cells to be in those specific places, right? We want our brain cells and our eye cells and our nose cells to be at the anterior end of our bodies. We want our feet at the posterior end, right? We want our spine to be on the dorsal side. We want our chest, our rib cage to be on the ventral side. So it's very important that we have this kind of spatial organization. And again, it's all of these signals that we've talked about before, the inductive pathways, the cytoplasmic determinants that help kind of organize the cells and be like, oh, okay, this is where I am. This is the things that I'm going to become. This is, maybe in different cells, like, okay, I'm over here. This is what I'm going to become. It's very important, especially for organisms that have bilateral symmetry. Bilateral symmetry meaning that there is one plane of axis that can we have symmetry. So for humans, we have one plane of axis down the middle where we are split. We are the same on both sides of that plane. And so we can kind of see what that looks like here. So here we've got the egg. It's kind of developing, right? It is unfertilized. We've got some depleted nerve cells. And my guess is that those nerve cells are actually releasing some signals because the anterior end of a fly, again, we're kind of looking at the Drosophila here as a model organism. The anterior end of the fly always starts off near those depleted nerve cells. So there probably is some induction going on there. But then we develop the different segments, and we can follow those green segments all the way to the development of the head. The brown segments become the thorax. The orange segments become the abdomen. So that's how we kind of get this whole pattern formation. Now, there are some specific genes that were identified that are play a significant role in the formation of these patterns, and they're called homeotic genes. And in Drosophila, they are crucial for proper larvae development, and really in all organisms. If we don't have this, the embryo is not going to develop properly. Some of them are called maternal effect genes, and that's because they come from the mother. So these are genes that are carried by the mother, not by the embryo. Now, the embryo has these genes, but whatever the embryo has, it doesn't actually affect the expression of them, or it doesn't affect the offspring itself. It's actually from the mother. And these can be known as egg polarity genes, and they control the orientation, which is another word for polarity of the egg cell. And one example is bicoid, which means two-tailed. So we're going to take a look at bicoid in a little bit more detail. So this is a normal larvae, and it has a head that is developing, and it has a tail. Now, what scientists did was that they mutated a whole bunch of larvae, or rather they mutated a whole bunch of flies, and then they bred those flies together, and they were looking for larvae that did not develop properly. And then they looked inside, and for some of them they found that there was a tail on both ends. And so you can see we have the A8, A7 segments that are connected to the A6 segment, and there's no expression of the anterior ends. What they found when they went looking is they found this bicoid protein was not present at all. Okay, so the bicoid protein is produced by the mother in an unfertilized egg cell, and it is concentrated at one end. Okay, and we find that mRNA is concentrated at that one end. And what happens is this creates kind of a gradient where you have one end of where there's a high concentration of the bicoid, and then as you kind of go through the larva, it becomes less and less and less. And this is what helps establish, because the bicoid protein regulates gene expression of the anterior, the head genes, the genes that are eventually going to become the head. So we have this kind of gradient that forms, and so we get the formation of the embryo properly. When that was knocked out, so if the mother had two mutant copies, there was no bicoid mRNA found, therefore no bicoid protein. So the larva was unable to develop the anterior end. So pretty cool, kind of an interesting thing where the mother actually influences the expression of the development of the fly in a much different way. So even if the fly had one mutant copy and one good copy, it still would not be able to develop properly. So these homeotic genes were studied a little further, and they were found in all organisms. So pretty much all animals have these homeobox genes, and in fact, we find them in fungi and plants as well. They're kind of shortened to Hox genes, homeobox Hox genes, and Hox genes have a 180 nucleotide sequence called a homeobox, and it specifies a 60 amino acid homeodomain within the encoded protein. So the Hox gene is much bigger than 180 nucleotides, but all of them have this specific 180 nucleotide sequence, and again, it has a specific 60 amino acid homeodomain. And that domain in the amino acids, right, in the protein, allows the DNA or allows the protein, whatever protein is created, to bind to any DNA segment, right? And so therefore, what we have is we have proteins that can bind to the DNA, and they can regulate the expression of different genes. And so variation in how the Hox genes are put together, the part outside of that 180 nucleotide homeobox, actually regulates different gene formation, right? So whether or not you have different sequences on either side of the homeobox, or if you have different expression of that Hox genes, that can actually lead to changes in the body plan. So if we take a look at a fly chromosome and mouse chromosomes, we find the same homeobox genes in both of them. We just find different copies of them, right? Or different amounts of them. So in the fly chromosome, we only have one copy of each, because if you might recall that flies only have 4 chromosomes. And so what we find is that these are in a specific order, and it leads to a specific body plan. Well, it's the same in the mouse. It's the same order of those genes, and that leads to a very similar body plan, right? Where you have the anterior end, and then you've got a posterior end. So the homeobox genes are extremely important in terms of the body plans, and very small variations in those Hox genes lead to all the different varieties of body plan that we see in the animal kingdom, right? We have so many different types of organisms and how they're all kind of put together, how they look, but all of that is due to variations in these specific Hox genes. And now there are a fair amount of them. There, it's not like there's just like 2 or 3 of them. There are a lot of different ones of them, but all of them are very similar. They're not very different. They're what we call conserved over time. They haven't changed very much. So pretty cool, kind of how the homeobox genes, those Hox genes work. And I believe that is all. Thanks for watching, and I'll see you next time.